Hello everyone and welcome to this lecture on new drug development and clinical trials, a very important topic. So we are going to gain knowledge of this today. So we are going to learn some interesting facts about some past discoveries, and then we will move on to the development of a new drug. Then we will learn that what is a clinical trial and its various phases. All these things are really very important to you all. So let's start with what knowledge do we already have? Well, the drug, we all know that it is defined as a chemical substance of known structure which, when administered to a living organism, it produces a biological effect. And we know that there are some really important drug regulatory authorities such as US FDA, that is US Food and Drug Administration. Then there is European Medicines Agency, and there is CDSCO, that is Central Drugs Standard Control Organization of India. So do you know this man? Well, he is Morpheus from the movie Matrix, and I think that his name was Morpheus because Morpheus is a name of God, and he was a Greek god of dreams. And one this Morpheus word is used in our pharmacology by Sir Turner. He was a German pharmacist, and he was the one who first to successfully isolate morphine from the poppy juice. This plant is known as opium poppy or Papaver somniferum, and he isolated morphine from its juice in around 1805 and named the substance morphium after this Greek god of dreams. This is morphine, and it has been long used for pain relief as an analgesic. Okay. Another interesting past discovery is that the women used to enhance their beauty by using an extract of a plant, and that plant was known as deadly nightshade or the devil's cherries, and its scientific name was Atropa belladonna, which means beautiful lady. Yes, and its name was Atropa belladonna because the women used to enhance their beauty because it contains an active constituent which is known as atropine, and this atropine causes the pupillary dilation, and pupillary dilation seems maybe seem as some beauty, so it was used like that. Okay. Next, you know this man, he is Alexander Fleming, and Staphylococcus aureus played an important role in the discovery of Penicillin G or benzyl penicillin, which saved millions lives actually. So, and he won Nobel Prize for this. So, as we all know that in the Alexander Fleming's lab, a culture plate of Staphylococci was contaminated by a mold. Later on, this mold was found to belong to the genus Penicillium, and it was eventually identified as Penicillium notatum. Now it is known as Penicillium chrysogenum. Okay, so these are some interesting past discoveries of some drugs, some very important drugs which are still in use. So, are you curious to know that how does a new drug is developed nowadays? Well, rather than these past discoveries we saw of morphine, of atropine, of penicillin G, today's new drugs are more invented than discovered. Okay, and this new drug development is time consuming process. It is an expensive process, and the overall cost to develop a new drug is around 1 to 4 billion dollars. So very expensive, a very huge amount. And this new drug development process starts from the laboratory, from the lab, and goes to the FDA approval. This whole process from the lab to the FDA approval. And after approval from the FDA, this drug is being marketed in the market, of course. So the whole outline of this new drug development is like this. It starts from the basic research in the labs. Okay, so in the basic research we do what? We find out many chemical entities. Okay, and once some promising new chemical entities are identified in this basic research in the labs, they enters into the preclinical tests in animals. Okay, so these new chemical entities which we find out in the basic research will be tested in the animals, and out of those new chemical entities, very few will be promising. Okay, and those promising new chemical entities will be later on tested in the humans, and when the human testing starts, it is known as clinical tests. Okay, and now the sponsor goes to the drug regulatory authorities such as FDA with all the data of human testing. Okay, and when the sponsor approaches to the FDA with all the data for the approval, if FDA finds out that the data is of good quality and it is of sufficient quantity, then it approves this new drug. Okay, and when the approval a sponsor gets, then it is marketed and the post-marketing surveillance starts. So this is the whole outline of a new drug development. Okay, so you can see that in basic research thousands of chemical entities are synthesized, then they are examined and then screened. So thousands of chemical entities are synthesized, examined and screened in the lab, and only a very few, almost 10 to 20, we find out that only 10 to 20 chemical entities are showing effect. Okay, and these new chemical entities, 10 to 20 new chemical entities, enters into the animal testing. Okay, and after animal testing starts the human testing. Okay, so in the human testing, number is decreasing, and when the human testing ends, only a single drug usually enters into the market after the FDA approval. Okay, so this is what is whole outline of a new drug development. So what do you think? That permission is required when a chemical entity is found promising in preclinical test, and then after that it goes to the clinical test being tested in the humans. What do you think? That permission is required for this jump? Yes, you are right. It is required because now human testing is being started, and if you want to test a drug in humans, you will have to get approval from the FDA. You will have to get permission from the FDA. Okay, and this permission you get by filing an application, and this application is known as investigational new drug application, or IND application, or INDA. This is a very important application. So the sponsor goes to the FDA with all the data which that sponsor accumulated during this preclinical test, and the sponsor goes to the FDA and files an application, IND application, and after that when he gets the approval, then only the human testing starts. Okay, this is very important, IND application. So after IND application is approved by the FDA, drug is given to the human volunteers. Okay, and you can see that this human testing is divided into three phases: phase 1, phase 2, and phase 3, while the post-marketing surveillance is named as phase 4. What are these phases? These are 4 phases of clinical trials. So the clinical trials are conducted in 4 phases. Phase 1 to 3 are designed to establish safety and efficacy. So we study how safe it is, this new drug, and its efficacy in human participants in treating the disease. When these 3 phases are complete, the sponsor applies to the FDA for approval to the market the drug, and we all know that. Yeah, so now just like IND application which was filed before phase 1 study, the sponsor will file another application after phase 3 study for FDA approval. Okay, and this application is known as NDA. Very important, new drug application, NDA. So FDA files this application after phase 3 with all the data of phase 1, phase 2, and phase 3. He goes to the FDA for the approval, and if it is approved, then this new drug enters in the phase 4. Okay, so IND application is filed before phase 1, NDA application is filed after phase 3. IND application with all the data of preclinical test, and NDA application with all the data of phase 1, 2 and 3. Okay. So after approval of the NDA, this new drug is allowed to be marketed in whole population, and post-marketing surveillance goes on to what? To do what? To gather additional safety data in larger population. Okay, we will see that. We will see first the phase 1 here. Okay, in detail. The phase 1 includes healthy volunteers. Okay, so healthy volunteers are required for the phase 1 study. So in phase 1 study, we do what? We take 10 to 100 healthy volunteers, and this study goes on for 1 to 2 years. Okay, 1 to 2 years. And what we do here is that we see this new drug, how safe it is, as well as we see what? How tolerable it is in these healthy volunteers. So we are not giving it to the patients in phase 1. We are just seeing that how safe it is and how tolerable it is. Okay, but there are some exceptions. Some exceptions are these, that if there is some advanced or rare disease and we can't wait, so what we do, we take participants with these diseases rather than healthy volunteers. Okay, so in phase 1 we can take some patients also, but only with advanced or rare diseases. It is an important exception. Next come the phase 2. So phase 1 is completed. The drug will go into the phase 2 of the study. So what we will see? We will see that in this phase, patient subjects are volunteered. We take some patients to receive this our experimental drug. So it is the first phase in which patients are included. In phase 1 there were healthy volunteers, and in phase 2 now we have patients. Okay, and we take almost 50 to 500 patients in this phase, and it takes around 2 to 3 years for this study. Okay, and what we see, as we have patients in this phase, we will see the efficacy of the drug, this experimental drug. Okay, so what we will see, that how effective this drug is in patients. Okay, as well as we will find out the dose of this drug, such as, suppose if it was paracetamol. Okay, so it may be founded in phase 2 that paracetamol's dose of 500 mg is effective in fever. So same goes like this in phase 2. We will see efficacy, how effective, and we will see the dose of the drug. Okay, so paracetamol is effective, we find out in phase 2, and its dose is 500 mg. We find out in phase 2 of the study. Okay, now comes the phase 3. So in phase 3, same thing, we have patients here, but the number of patients is quite larger. In phase 2, we have around 50 patients, while in phase 3, we have a few hundred to few thousand participants. Okay, so it is usually a multi-site. Okay, so in phase 3, we hire multiple sites and we take participants in these multiple sites and we study the drug, and it takes around 3 to 5 years. And we already saw the efficacy in phase 2. What we do in phase 3, we just confirm the efficacy, how effective the drug is in our larger population. Okay. And after all these phases, phase 1, phase 2, phase 3, with all these data, a sponsor goes to the FDA, and if FDA approves, this experimental drug will go into the market. And when it enters into the market, still we have eyes on that drug, and this is known as post-marketing surveillance. Okay, of course we will give it to the patients, but in what? In a normal condition in our clinics. Okay, and of course we will give to many participants, many patients. Okay, and there is no fixed duration of this. What we are going to see, we are going to see the long-term safety of this drug. We will see if it causes any adverse event. We will see in this normal condition, suppose paracetamol, we are giving it to the patient in a normal situation, but with fever, patient is also suffering from suppose diarrhea, and we are giving him or her some anti-diarrheal drug also. Okay, so we will see the interaction between these drugs, these anti-diarrheal drugs as well as paracetamol, what are their interactions. So all this we study in the phase 4. Okay, that is what will happen in the post-marketing surveillance. So I have accumulated all that data in this table. So phase 1, first in humans, in healthy volunteers, occasionally in patients with advanced or rare diseases. There are 10 to 100 participants. It takes 1 to 2 years. It is to know how safe is the drug and how tolerable the drug is. Then in phase 2, first in the patients will receive the experimental drug. We will see here that there will be 50 to 500 participants. It takes 2 to 3 years, and we will see the efficacy, how effective the drug is and what its dose should be. And the phase 3, in phase 3 it will be a multi-site trial because we require many participants, and it goes for 3 to 5 years, and we just confirm the efficacy. And in phase 4 it is post marketing, after approval of the drug, and we will give it to many patients, and there's no fixed duration. We will see the long-term safety. We will see if there are any adverse events of the drug which we did not find out in these 3 phases, as well as we will see some drug interactions if there are any. Okay, so these are 4 phases of clinical trials. Okay, very important. I hope that you all got it. So the knowledge we gained today is that clinical trials are conducted in 4 phases. Phases 1 to 3 are designed to establish safety and efficacy of this experimental drug. After its approval, comes the phase 4, that is post-marketing surveillance for additional safety data. And now we know that drug is developed, and its process starts from the basic research in the lab, then the preclinical tests in animals, then the sponsor files for the IND application, then the human tests start from the phase 1, then it goes to the phase 2, then phase 3, and after this phase 3, sponsor again goes to the FDA with NDA, new drug application, and when this new drug is approved, it goes into the market, and the post-marketing surveillance continues, and it is known as phase 4. These are the references. Now you have assignment. What you are going to do is that nowadays a new phase has been added in clinical trials, and it is known as phase 0. Okay, although it is not compulsory to perform this phase for FDA approval, but sponsors still may perform this phase. So you have to tell me what is this phase 0, as well as another question is what is new chemical entity. Okay, share this in the comment section. Thanks a lot. Do subscribe, like, and share. Thank you.